All right, everyone, we made it to the end of week 6. I tried to get out of my office on Friday afternoon before it started raining, but it's starting to come down. Just wanted to bring you a little update in terms of some of the AI-generated resources I'm providing for you all. So a lot of the topics that we spent our time on, right? The electron transport chain, chemiosmosis, cellular respiration in general. I have a really great AI-generated podcast that goes over it. So you will be able to watch it at the end of this video, or I'm going to upload it as like just an audio file that you can find on D2L. So go check that out from the announcement, download it, listen to it. I think it's a really good review tool and just something that will help you think through some of these processes a little bit later. The other thing that I'm doing is when you look at the D2L announcement, I'm actually going to have like 10 additional study guide questions. These are also AI generated based off of the transcript that I provided to the chatbot, but they're there for you just to give you a little bit more practice. I recommend just going through them, like take 2 to 3 sentences to fill that stuff out, see if you really understand that material. And if not, you can come bring that to me during office hours, come see me at the beginning of class, we'll go take a look at it. All right, so I'm gonna just like try to book it to my car. Again, right after this, you'll see the AI generated podcast. Hope you enjoy it. See you all next week.
All right, let's dive into this amazing world of cellular energy. You sent us this incredible lecture and we're gonna break it down. Think of us as your guides to the inner workings of how our very cells make energy.
It's kind of like going behind the curtain at a magic show, except it's the real magic of biology.
I like that. We're diving deep into glycolysis, and then the citric acid cycle, then the electron transport chain, the whole 9 yards. But before we get lost in the details, can you remind us where all this energy production is actually going down?
Absolutely. The main stage for this cellular performance is the mitochondria. It's often called the powerhouse of the cell for a reason. Like, picture a bustling factory. That's where our cells take glucose, simple sugar from the things we eat, and they transform it into, the energy we can actually use.
Okay, glucose as fuel makes sense to me. But how does it actually become the kind of energy our cells can use? I'm guessing it's a bit more complicated than just plugging the glucose into a wall socket.
Yeah, it's way cooler than that. It all starts with glycolysis, which literally means sugar breaking. It's like Act 1 in our energy production story.
Glycolysis is the opening act, okay? So what's going down during this very first step?
Imagine a big pile of lumber, that's our glucose, and you have to break it down into smaller, more manageable pieces before you can build anything with it, right? Well, glycolysis is like taking that first step. It's breaking down that glucose into these smaller molecules called pyruvate.
Okay, so we're breaking down glucose into pyruvate. Got it. But breaking stuff down, that takes energy too, doesn't it? So where does that initial energy even come from?
That's a great question. So glycolysis has these 2 main phases. There's the investment phase and the payoff phase. In the investment phase, the cell uses a tiny bit of its own energy currency. That's ATP, think of it like cellular cash, to kickstart the whole process. It's like spending a little to make a lot more later on.
I'm listening. Tell me more about this payoff phase then.
All right, so during the payoff phase, the process generates more ACP than it actually used in that initial investment phase. It's like investing in a stock that doubles in value. We end up with a net gain of 2 whole ATP molecules.
So our cells are like expert investors then. They get a return on their energy. But what happens to those pyruvate molecules that get created? Now, are there any other, important players in this glycolysis process we should know about?
You're on a roll. Remember how we're trying to get energy from glucose? Well, during glycolysis, we also make these cool little molecules called NADH. They're like tiny energy-packed batteries, and they get charged up by grabbing electrons from the glucose.
So NADH is like an electron shuttle then. It grabs those high-energy electrons and hangs onto them for later.
You got it. These electron shuttles are key because they come back later in the whole energy production process. Now, as for the pyruvate, it's still full of potential energy, but it has to be prepped and transported to a different part of the cell before we can unlock all that energy.
Wait, a different part? I thought we were hanging out in the mitochondria, the cell's energy factory.
We are. But glycolysis actually takes place outside the mitochondria. It happens in the cell's cytoplasm. It's like the prep work before the main event.
Gotcha. So we've prepped the fuel source, charged up some electron shuttles, and now our pyruvate is all ready to go into the mitochondria for the next phase.
Ha! This is getting good. What's next on our little cellular energy adventure? So our little pyruvate molecules are all dressed up with somewhere to go. It's time to enter the inner sanctum of the mitochondria. What's going on when they arrive?
Get ready for the main event. Inside the mitochondria, pyruvate goes through these two key processes. First up, pyruvate oxidation, and then we move on to the citric acid cycle.
Okay, two big names. Let's break them down. Pyruvate oxidation first. What's actually getting oxidized here?
Basically, we're getting that pyruvate ready to enter the citric acid cycle, like giving it a VIP pass to the party. And during this process, pyruvate loses a carbon atom. It gets released as carbon dioxide. Hold on, carbon dioxide, you mean the stuff we breathe out? Are you saying we're literally exhaling a part of this energy conversion process?
You got it. It's all connected. And there's even more to it. Remember those NADH electron shuttles we were talking about from glycolysis? Well, in pyruvate oxidation, another NAD molecule comes in, grabs an electron, and becomes, you guessed it, NADH.
I'm really charging up those energy batteries.
Yep.
Okay, so pyruvate is prepped, more electrons are stored away.
Yeah.
Bring on the citric acid cycle. What makes this cycle so important?
Picture this. The citric acid cycle is like a master extractor. It's like the main job is to squeeze every last drop of usable energy from what's left of our original glucose molecule.
That's efficient. How does it pull that off?
Well, through this super cool series of chemical reactions, each one is catalyzed by a very specific enzyme.
Hold on. Can we rewind for a second? Can you explain what catalyzed by an enzyme actually means for those of us who, need a little refresher?
Of course. Enzymes are basically like tiny biological machines, and their job is to speed up those chemical reactions. Picture a factory production line, right? Each enzyme is a specialized worker, making sure that their step in the whole process happens quickly and without any mistakes.
Gotcha. So enzymes are the expert workers in this cellular factory. Okay, back to the citric acid cycle. What's going on in this production line of ours?
As molecules move through the cycle, they get rearranged, they get broken down, rebuilt, you name it. And during these transformations, we're producing even more of those electron carriers. We get NADH and a new type called FADH2.
Two different kinds of electron carriers. It's like our cells have a diverse energy portfolio or something.
Exactly. Diversity is key in this process. Now, while the main goal of the citric acid cycle is to load up those electron carriers, there's a little bonus round here, we also get a little bit of ATP made directly during this process.
Direct ATP production, I'd love to hear it. But you're making it clear that the real payoff comes later, right? From all those electron carriers we've been stockpiling this whole time.
You're catching on quickly. Those electron carriers, all loaded up, they're like delivery trucks heading to their final destination, the electron transport chain. And that's where they finally cash in all that precious energy cargo.
Okay, I am officially curious about this electron transport chain. It sounds like the grand finale of cellular respiration. Hit me with it. What happens there? All right, time for the grand finale. We've made it to the electron transport chain. This is where those electron carriers we've been talking about finally cash in. And our cells, well, they hit the energy jackpot. I'm ready. Take me there.
Okay, picture this. You've got a line of proteins, and they're all embedded in the mitochondria's inner membrane. That's our electron transport chain. It's like, a cellular conveyor belt? Or maybe a bucket brigade is a good way to think about it.
I like the bucket brigade. Each protein passes something along to the next, right? So our NADH and FADH2, they roll up with their buckets full of electrons. What happens next?
They hand off those electrons, those high-energy electrons, to the proteins in the chain. And as those electrons get passed down the line from one protein to the next, something really cool happens. They release energy.
Oh, so the electrons are giving off energy as they move down the chain. So what happens to all that energy being released? We don't want it going to waste.
Definitely not. This is where things get really, really smart. The energy from the electrons, it's used to pump protons, which are these positively charged particles right across the membrane. It's kind of like using a water wheel to pump water uphill.
Okay, so we're pumping protons uphill. I'm seeing a pattern here. Our cells are really good at building up potential energy. What's the strategy with this proton pumping? Why is it so important?
Well, by moving all those protons to one side of the membrane, what we're doing is we're creating this proton gradient. It's a difference in concentration. You can think of it like water building up behind a dam. And just like a dam, which can release this powerful flow of water, that proton gradient is storing a ton of potential energy.
Okay, so we've built up this proton dam. It's ready to burst. How do we actually capture that energy? How do we convert it into ATP, that usable energy our cells need?
That is where ATP synthase comes in. It's like the cell's very own hydroelectric turbine. This incredible protein is actually embedded in the same membrane, and as those protons flow back down their gradient, they pass through ATP synthase. And guess what? They make it spin. That spinning action, it generates ATP from ADP and phosphate.
Hold on. It's like the turbine spinning and generating electricity in a dam. But here, we're generating ATP. That's wild. We started with this one glucose molecule, and look how much energy our cells can get from it.
It's truly remarkable. This whole intricate, elegant process is what we call cellular respiration. We take our energy for granted, but there's so much going on under the hood, inside every single cell of our bodies, every single second of the day.
This deep dive has given me such a new appreciation for what's happening inside my own body. I don't think I'll ever look at food the same way again, that's for sure. Knowing that all those cells are going to be busy breaking down those glucose molecules and making energy, it's pretty amazing.
It really is. It just goes to show even the most complex processes can come down to these elegant, fundamental principles. And that's the beauty of biology.
Absolutely. And on that note, we'll leave our listeners to ponder the wonders of cellular energy. Until next time, everyone, keep exploring, keep asking those questions, and keep diving deep with us.